First of all, I would like to thank organizers for considering to give this opportunity for us. It's a honor to be here talking to these experts around here. So especially I would like to thank Mr. Sharad sir, Sharad Deshpande sir. He was very instrumental in facilitating my talk. So in the next 15 to 20 minutes, what I'm going to do is walk you through something different than what you have heard for the last 2 days. So I'm going to be introducing you to some new technology that is using seed plant. So basically these are nothing but seaweeds. Since we grow them, we call them as seed plants. So we have made a few products and these products make a difference to the crops at the gene level. So that is the relevance I am giving this talk here. As the topic of the presentation is in the evolution for profitable agriculture, we have the products that make impact on the genetic level, at least transiently, and thereby lead to a positive or beneficial effects in the crops, whether it's with respect to the yield or with respect to the defense. So before going into the talk, I'll just briefly introduce about my company. We are purely and proudly made in India company. We are started sometime in 2010 from the campus of IIT Chennai. A group of grad students guided by Dr. Shikumar Suryanarayan, who was the director for back on R&D for the last 25 years. From ever since it started from Chennai campus, immediately the company moved to the center of biotech cluster in India, that is NCT's campus. So currently, in the last 7-8 years, we've been placed at Center for Cellular and Molecular Platform, Bengaluru. So the idea of the company is making ocean farming. The goal of the company is ocean farming. So what do I mean by that? We want to use seaweed biomass, that is peatland biomass that we cultivate, to make the products that can make a huge impact with respect to food, feed, and fuel aspects. So basically, we have technology for making a a large amount of seaweed biomass. This is in-house technology that we have developed. And we have a lab where we can convert this seaweed biomass into value-added products. And that can make impact in the area of food sector, feed sector, and fuel sector. So with respect to the biomass production technology, we have developed mechanized versions of seed tractor. This is called seed combine. So this can go on the sea surface and seed the seaweed plants, and it can also harvest the seaweed plants. And this is the deep technology that can give us accessory support to the seed tractor. Basically, it can help us generate the solar power, and also it can help us provide nutrients by upwelling the nutrients in the sea underneath. So that way we have this technology that can make us very powerful in growing a large amount of seaweed biomass that we need for making the products in this area. So in the lab, that's in the bioconversion lab, we are very 10 years old company. We have already started to make products in various areas. So we already have POC type products in animal feed area, fine chemicals, biofuel, and also the food additives. We have made a large progress with respect to agri-related products. We have made remarkably good success in this area of product lines. So in the interest of today's talk, I am going to further restrict myself to the agri-related products that we have. The use of seaweed in agriculture is not something new. It has been there historically. People have used the dried seaweed powder as a fertilizer. the coastal farming community. But largely, there are so many products that are based on the brown seaweed. The brown seaweed, just for those who have less knowledge than seaweed, there are three classes of seaweeds: brown seaweed, red seaweed, and green seaweed. So based on the pigment they have. Uh, the red seaweed can be cultivated, whereas the other two seaweeds are mostly grown in a wild situation. They are naturally grown. So people have caught up on this feature. They generally used brown seaweed for making the extract and call them as a biostimulant product. So if you look at the literature, there is a lot of literature which supports the effect of these products, biostimulant products, on plant growth. So as I said in the beginning, what we do is we use the cultivated red sea plant, that is Capophycus species red sea plant. We use this biomass, and with respect to agri products, we have made two line of products. First one was this is the first product the company made, biostimulant line of product. It is sold under the name AgroGain, and so this product basically it influences when we apply it on the foliar, it improves the photosynthesis rate and also increases the nutrient uptake from the soil, thereby reflecting in the higher yields. And the second product which we have recently made is a biodefense line of product. So these products are focused on improving the plant health. So basically, both this category of products, when we apply on the foliage, they tend to enhance certain genes responsible for these activities, thereby leading to a transient kinetic effect. And so those effects can be positively seen with respect to other fields or with respect to the plant health. So I am going to walk you through in next 15 minutes or so, I am going to walk you through the data on both these products. The first one, AgroGain, as I said, it's a biostimulant product. So this is basically a very natural product. So this is prepared using the fresh cultivated red sea plant. So it is unlike other products that we have in the market, they all use the dried seaweed powder for making the extract. So this is unique in that aspect. And this product we recommend apply on foliage at 1 ml per liter. And this is a patented formulation from us. And it is a purely organic product certified. We also have understood the mechanism of action of this product. So at the outlook, these are the phenotypic changes the product offers. So you can clearly see the increase in root proliferation as well as the shoot growth and also leaf area. Basically, it does all this by enhancing the cell division effect. So I'm going to go through the gene candidates that are replaced in this. So before that, I'll just show some lab data. This is the bioassays that each of the product batches that we make in the lab go through these assays as a part of maintaining the quality of the product batches. So what you see on the left is the control group and on the right is the treated group. You can clearly appreciate the difference between the two groups. And similarly with other crops as well, the rice, tomato, maize and tobacco, you can appreciate the difference in the root mass and the shoot length. So basically it is working through increasing both the root mass and the shoot length. And then this is the data to show there is no added phytohormones in the formulation. We have not added any chemicals. We are not fortified with any of the minerals. This is purely what is there in the sea plant. It's totally natural. And we don't even use harsh acid solutions in our process during the preparation. So you can clearly see here the phytohormone levels are far too less to detect in our products. And basically this analysis was done on the stock product. We use this product at 1:1000 dilution. So whatever these numbers we see here, they should be diluted further, divided further by 1000 by the time it goes onto the plant. So there is hardly any phytochemicals, phytohormones that are there in the product as such. But what they do, once you apply this product on the plant, it actually influences some of the hormone levels to go up. So that way it brings up the effect. We have done the trials quite extensively. We have ventured in more than 14 countries, and we have done 40 plus institutional trials, and we have done 100 plus R&D trials across 20 different crops. And these are the pointed regions where the studies, some of the studies are going on, some have been concluded. And this is the data, the final trial result summary from the trials, just briefly. This is presented against the regular, the increase, percent increase in the yield over POP, that is regular practice. So you can see up to 14% increase in yield in case of onion, and highest we see about up to 30-35% increase in case of crops like chili and potato. So it makes a remarkable difference with respect to final output. And in order to, since ours is a purely science-driven company, and we are there in such atmosphere between CKVK and NCDS campus, we tend to understand what's the science going behind this. So we have conducted transcriptome-based work, and this is the data heat map showing the 48-hour post-application of the product. This is on the foliar tomato foliage. You can see on the untreated group, and this is the treated group. You can clearly see the redness, which is a higher level of gene expression. And important candidates are mentioned here. Some of these genes are related to flowering. Some of them are related, as I mentioned earlier, to cell division and growth and metabolism. And a few are related to the capturing light, this photosynthesis activity. And here is the mapping of the genes to the pathways. You can clearly appreciate that greater than twofold difference. That is, some of them are at least 200-fold increase in the gene level expression you can appreciate. And a lot of these genes are related to the phenotype difference that we notice in the graph. So just to summarize on this part, so this is a purely organic product, and it is highly scalable and scalable, and this is a product that can actually fit into our sustainable practice of agriculture. And we have seen always a consistent effect that is even low as like 10% or even high as 30-35%. We have seen a remarkable difference with respect to yield when this product is used in the foliage. And we do understand the mechanism of action, so there is always a room for making better versions in the coming days. So now I am going to switch gears and completely talk about another line of product, which is the biodefense line of product. So as we all know, even my previous speakers also have stressed upon what is the kind of biotic stresses the plants have to go through, the crop plants have to go through. The viruses have been a big menace, right? So there are different numbers depending on the source that we look at. And one that has attracted my attention is this one. You know, there is about $60 billion loss in crops due to the virus menace across the world in each year. And interestingly, Professor Muniappa, way back probably in '73, had published one paper in '89. '89 had published a paper on tomato TOLCB infection rate, and in the earlier talk, Prashant Belgamer also alluded to this fact. If we can protect the crop for first 45 to 50 days, the danger is going to be, the danger and the loss is going to be minimal. So Professor Muniappa had very elegantly described this in the paper, where he had shown with respect to TOLCB infection. If it occurs before 20 days, loss up to 90% or greater can be expected. But when it occurs around 35 days, the loss can be accounted for up to 74% of the crop yields. Whereas if it occurs after 50 days, it can be as low as 20%. So this is the idea that we are trying to catch on. We are trying to make a product that can protect the plants for initial 50 days, at least till the plant enters into the reproductive phase. So based on that idea, we are trying to make the products using this red seaweed biomass. And unfortunately, this is known to all of us. The farmers don't have much options. Of course, builders have made several progress in this making the distant lines, but each of the lines have certain specific life, so most of them will susceptible. And a lot of the time, farmer end up using excess of chemicals. That's the only means controlling the insecticides. So the other new technologies are always welcome in this area. So that's where we jumped in, and then we thought whether we can make any difference to this area. So we have formulated this solution. Actually, I had a picture of the bottle and that the product formulation. Some reasons it's not coming out. So this product, what we developed is a daily food. This is again a completely natural, sustainable, eco-friendly product. This basically works on the plant immune system. So we all know the plants do have a system to fight off the pathogen. When the virus enter, there are already existing mechanisms in the plant. We want to capture upon them, boost them up, and see if that alone can be sufficient to ward off the virus. So this is the approach we have been taking. So idea here is to have the product used in a prophylactic approach. That is not as a curative, like not after getting the disease, but once we plant into the field, we recommend the farmer to start using it at least every 15 days once. And this product we have tested quite rigorously with various universities. Again, we have some idea about the mechanism of action, which I'm going to walk you through. So this is the fundamental assay that we use for developing this product. So this is a very simple assay. Papaya can be easily infected with papaya ring spot virus. And papaya ring spot virus is a very deadly disease, and this is papaya ring spot virus disease almost is regarded as a cancer of humans. Once it occurs in the field, there is hardly any way to control it, contain it. So in this assay, what we do is we have two sets of plants. First, the treated group are primed with our product at 1.5 ml per liter, and then on the next day, we apply the SAP, the TRS inoculum, and then look at the virus load using the oligoprobe in a qPCR-based assay. So interestingly, when we looked at the virus load, that is the RP gene of the virus, we could clearly see a big difference, at least 50% difference with respect to the virus load.
1 more minutes left.
Okay. So, and so that gave us a very good promise, and we left the plans to go for further days, and we could capture some interesting phenotypes. We could see the severity is pretty high, the rat having the symptoms very high in case of untreated group, whereas in case of treated group, the symptoms were there. It was not like zero, but there were symptoms, but they were just in the beginning. So there is a delay in the onset of disease. So we did trials as well around North Bangalore, and these are the field pictures. And what we noticed in these trials is there is a difference with respect to the fruit setting as well. There are the less symptomatic fruits and less ambulation on the fruits, and there is more number of fruiting. And interestingly, what was important for us is to look at the blue line and the orange line. So the orange line is the untreated group and the blue line is the treated group. There is a difference in the disease onset. So these studies were carried out with Professor Nagaraj, plant pathology, here GKVK, and in fact based on these studies, the university has proposed a technology for managing the papaya ring spot virus as a package of practice in the integrated management of TRSP. We also have done tomato trials. Again, these were done at the hotspot regions, where these diseases are common. And we clearly see a difference. It's not like there is no incidence, there is an incidence, but the severity is low and the percentage of incidence is also less. So the product is indeed making a difference, and in the current situation, it is making a very significant difference. So in order to further understand the mechanism of action, we further went into the transcriptome studies. We clearly understood there are genes such as salicylic acid phosphorylated genes and also PAL and other stress-related genes which have gone several-fold up. And based on this information, we have put together this hypothesis wherein we see the binding of ingredients at the cell surface results in a calcium burst. We see a lot of calcium-dependent colmodulin domains go up. And then we also see the PAL going up with respect to expression level. And then we also see the SABP gene going up several fold in our transcription data. So SABP is a gene that the protein that hydrolyzes methyl group. So methyl salicylate is the transport form of the salicylic acid, whereas just the salicylic acid, once it hydrolyzes the methyl, it brings up the action. So that's where the SABP, once it goes up, there is more of active form of SA, so leading to the further downstream effects. So just to quickly summarize, this is again another natural product. It can be very well fitted into the integrated management approach. Of course, not standalone, but definitely in the integrated management approaches. And quickly, I will walk you through these 4 slides where we have touched upon another interesting product in the lab. This is the product we refer as LPD, still in the R&D pipeline. And this product, very interestingly, doesn't have a direct effect on a fungal pathogen. So we look at the plates here, we have included in the plate our product, and this is the control of the growth, but the fungus is comparable, whereas in case of a fungicide it is reduced. So it doesn't have direct effect on a fungus, but when you throw it under the plant, it induces certain genes. They are called, like the 2 of them I shown in a PDF 1.2, VSP2, going up to 4 to 5 fold here. We clearly see these genes are related to the antifungal pathway. In fact, this is in the NCBI database, this gene is related to J-related pathway or antifungal gene. So definitely there is a talk happening with respect to product application. There is influence on the J-related pathways. This is another cycle cool picture. So this is looking at the detachment facet, wherein we apply the product on the whole plant, collect the leaf and put the spores, inoculum, alternaria spores on the center of the lamina, the leaf lamina, and incubate over 10 to 15 days. You can clearly see in the untreated group the fungal lesion area going up, whereas in case of the product treated the lesion area is contained to the tissue region. And this is the estimate of the scan images. We have also done the field trials with respect to containing the tomato blight, severity again with respect to with the collaboration with Girish T R. And we have observed our products application alone and foliage and independently can give protection at least on par with the fungicide. So if not, the product can be used independently. At least it can be used as an interval as alternate treatment to fungicide. Thereby, we can reduce the fungicide, residual fungicide in our food. So this is where I am going to end with. The products that I will walk you through are purely natural, and this is a unique approach that we have taken to. And these are completely science-driven products, technologies. Idea is to facilitate the integrated management approaches by bringing in this kind of natural products so as to reduce the chemical usage or chemical footprints in the environment and also the food that we consume. This takes a good context in the background that Professor Gowda mentioned yesterday. There is too much of fasting on the breeder, but this kind of yesterday you mentioned for some Q&A questions. So these kind of products definitely help the farmers where the breeder efforts are taking time. Thank you very much for your attention.
Thank you for your presentation, especially for what we call green agriculture or organic based agriculture. So maybe if there are any queries, questions.
Hello, Dr. Girish, thanks for the nice presentation. I have only one question. Do you have anything for the seed treatment?
Sir, seed coating. We are trying to soak the seeds and see once we sow them and look at the lab on the white flies with respect to susceptible varieties. Once we grow the plants with the seed coating and then infect those plants, see whether there is a difference in the symptom severity or not. So we have seen initial results to suggest very interesting results. So we have used in one of the latest assays, Election 22, as the tomato plants for inoculating using white flies, inoculating ULCV. We have seen very interesting results. So, but again, these are in the R&D pipeline. So probably by in few months we will have want to share it. Yeah, we definitely have thoughts in that direction as well.
Thank you.
Preventive or curative? Virus.
It is preventive, I want to say. The curative is the overstatement which I make, sir.
Yeah, that's what I have seen that paper by
Right.
Dr. Nagaraj and et al.
Right, sir.
So it is combination of many agricultural practices
Right.
like mulching and
Yeah.
everything,
Yeah.
and then or a mixed cropping with banana and all that you have done.
Yeah, that's great if somebody can do that, but lot of the farmers that I visit, they don't even put the barrier crops. So what they do, for them that the integrated management means just use different chemicals. So what they do, that is they use the insect control, something like Candida, they use Demeter,
Yeah.
and in that aspect, even if they combine the insecticide and our product, they are working on two different aspects, two different subjects. One is working on plants, other one is working on the pathogen. So it is still something to say as an integrated aspect. But as you said, that's the ideal case. If somebody can put the barrier crops or even the intercrops, that's a great thing. Or silver mulch, that's a great thing to do. That's what we stressed about in the paper.
So once it is infected, the plant once it is infected, it cannot be cured.
So the notice is, it doesn't put the same disease pressure on the plant as the untreated group. So there is a difference. Even though the plant is diseased, it does produce the yields, but the symptom severity is lower. So that way, one can still expect some type of an income if they start using these products.
Okay. How many kgs per plant you got?
I can share the details with you. It's not at the back of
We
my
can meet
mind.
up during your.
Okay, sure.
All right.
Here and now. Yeah.
Yeah, there is a question at the back, maybe can you, yeah. Give them.
I have an uncle who is an agro input dealer. We have come across this product and farmers are aware of the seaweeds, some biostimulants and protein lysates. The use of these has been increasing now, and they are aware of the like biofarming or something like organic farming. They are somewhat stepping backwards using the chemical products. So they are aware of these products. And are there any products coming out to use them as herbicides?
No.
We have seen across only in plant stimulants, growth stimulants,
We
yielding
don't have
enhancers.
weedicides or herbicides yet.
They are not
Not
biomolecules.
Yeah, no.
So thank you.
Thank you. So maybe we can request to Mr. Sukhdare to